Hello guys, welcome to Education Era. Today I am going to explain you the recombinant DNA technology. And this is the content of recombinant DNA technology. First I will explain history of recombinant DNA technology, definition of recombinant DNA technology, the steps of recombinant DNA technology, and under the steps first, restriction enzyme digestion, amplification using PCR, ligation of DNA molecules, insertion of recombinant DNA into host, isolation of recombinant cells, and then last, application of recombinant DNA technology. The history of recombinant DNA technology. The foundation of recombinant DNA technology was laid in 1970 with the discovery of a group of restriction and nuclease enzymes in microRNAs by Hamilton Smith. In 1973, Herbert Boyer and Stanley Cohen successfully recombined two plasmids, PSC-101 and PSC-102, and cloned the new plasmid in E. coli. E. coli is a bacteria. Plasmid PSC-101 possesses a gene resistant to antibiotic tetracycline, while plasmid PSC-102 possesses a gene resistance to antibiotic kanamycin. The newly developed recombined plasmid, when incorporated into the bacteria, exhibited resistance to both the antibiotics tetracycline and kanamycin. After this protein, somatostatin in 1976, insulin in 1978, growth hormone in 1979, and alpha interferon in 1980 were produced by recombinant DNA technology. Definition of recombinant DNA technology. The definition of recombinant technology is recombinant DNA is produced by recombinant DNA technology. Formation of recombinant DNA is usually done by combining two or more DNA segments of different organisms. In the recombinant DNA, so formed genes of one organism are combined with a regulatory sequence of another organism. Such type of gene is called chimeric gene. Chimera is a monster in Greek mythology that has a lion's head, a goat's body, and a serpent's tail. This is comparable to Narasimha in Indian mythology. Recombinant DNA is produced for any of the following purposes. First, to obtain multiple copies of our DNA segments. Second, to integrate a specific gene in the chromosome of an organism for producing the concerned specific character. Third, to obtain ample amount of protein produced by a specific gene. Steps of genetic recombinant DNA. First, isolation of genetic material. The first step in recombinant DNA technology is to isolate the desired DNA in its pure form, that is, free from other macromolecules. Since DNA exists within the cell membrane along with other macromolecules such as RNA, polysaccharides, protein, and lipids, it must be separated and purified which involves enzymes such as lysozymes, cellulase, chitinase, ribonuclease, and proteinases, etc. Other macromolecules are removable with other enzymes or treatments. Ultimately, the addition of ethanol causes the DNA to precipitate out as fine threads. This is then pulled out to get purified DNA. In detail, we explain a diagram. In this diagram, cells are lysed using a detergent that disturbs the plasma membrane. In second step, cell contents are treated with proteases to destroy protein and RNase to destroy RNA. After that, cell debris is pelleted in a centrifuge. The supernatant liquid containing the DNA is transferred to a clean tube. The DNA is precipitated with ethanol. It forms viscous strand that can be spooled on a glass rod. Second step, restriction enzyme digestion. Restriction enzymes act as a molecular seizure. that cut DNA at specific locations. These reactions are called restriction enzyme digestions. They involve the incubation of the purified DNA with the selected restriction enzymes at condition optimal for that specific enzyme. The technique agarose and the restriction digestion, the scissors cut two ends, sticky end and blunt end. The technique agarose gel electrophoresis reveals the process of the restriction enzyme digestion. This technique involves running out the DNA on an agarose gel. On the application of current, the negatively charged DNA travels to the positive electrode and is separated out based on size. This allows separating and cutting out the digested DNA fragments. The vector DNA is also processed using the same procedure. This is our target DNA and endonucleases cutted our target DNA ends. This is sticky ends and this is our electrophoresis. This is our gel cascade filled with buffer solution. This is the direction of our DNA movement. These are the electrodes, anode and cathode. This is the sample wells. Those were filled with our DNA extract. Third, amplification using PCR. Polymerase chain reaction or PCR is a method for making multiple copies of a DNA sequence using the enzyme DNA polymerase in vitro. It helps to amplify a single copy or a few copies of DNA into thousands to millions of copies. PCR reactions are run on thermal cyclers using the following components. First, template means DNA to be amplified over target DNA. Primers, small chemically synthesized oligonucleotides that are complementary to a region of the DNA. Enzyme, DNA polymerase, nucleotide needed to extend the primers via the enzymes. And the cut fragments of DNA can be amplified using PCR and then ligated with the cut vector. This is the figure showing PCR is polymerase chain reaction. Polymerase chain reaction completed in 3 steps. This process completed in 3 steps. First, denaturation, second, annealing, then extension. Denaturation means separate our double-stranded DNA. Annealing means anneal the primers, and then extension means elongation of our DNA. Ligation of DNA molecules. The purified DNA and the vector of interest are cut with the same restriction enzyme. This gives us the cut fragments of DNA and the cut vectors that is now open. The process of joining these two pieces together using the enzyme DNA ligase is called ligation. The resulting DNA molecule is a hybrid of two DNA molecules, the interest molecules and the vector. In the terminology of genetics, this intermixing of two different DNA strands is called recombination. Hence, this new hybrid DNA molecule is also called a recombinant DNA molecules, and the technology is referred to as the recombinant DNA technology. This is our plasmid and this is our target DNA. Both are mixing and joined with the enzyme DNA ligase and will make our recombinant plasmid. Insertion of recombinant DNA into host. In this step, the recombinant DNA is introduced into a recipient, host cell mostly, a bacterial cell. This process is called transformation. Bacterial cells do not accept foreign DNA easily. Therefore, they are treated to make them competent to accept new DNA. The processes used may be thermal shock, Ca++ ion treatment, and electroporation, etc. Sixth, isolation of recombinant cells. The transformation process generates a mixed population of transformed and non-transformed host cells. The selection process involves filtering the transformed host cells only. These cells we transfer into the host cell. For isolation of recombinant cells from non-recombinant cells that we do not transfer it, marker gene of plasmid vector is employed. For example, PVR322 plasmid vector contain different marker genes like ampicillin resistance gene and tetracycline resistant gene. When PST1 restriction enzyme is used, it knock out ampicillin resistance gene from plasmid so that the recombinant cell becomes sensitive to ampicillin. This is our recombinant DNA plasmid. We transferred it into E. coli, a bacteria. This is our recombinant DNA plasmid, and this is the DNA of bacteria. And this is the ampicillin plate cells onto agarose gels containing ampicillin. Expand the positive clones to exponentially replicated copies of desired DNA. Then isolate DNA. Then test biological properties of your gene of interest and show the gene expression. This diagram also shows what before we explain. And last, application of recombinant DNA technology. Recombinant DNA is widely used in biotechnology, medicine, and research. The most common application of recombinant DNA is in basic research, in which the technology is important to most current work in biological and biomedical sciences. Recombinant DNA is used to identify, map, and sequence of genes and to determine their function. Recombinant proteins are widely used as reagents in laboratory experiments and to generate antibody probes for examining protein synthesis within cells and organisms. Many additional practical applications of recombinant DNA are found in industry, food production, human and veterinary medicine, agriculture, and bioengineering. And this is our reference where I make my PPT. Thank you so much, and this and thank you so much. Please like and subscribe my channel. If you have any query, then please comment it. I will explain in other video. Thank you.